जैसे कि लास्ट क्लास में हम in the last class we were discussing about the primary, secondary, tertiary as well as the quaternary structure of the protein. Quaternary. So primary is basically a sequence of amino acids. So it has no chain. So for example, if we write alanine, valine, threonine, glycine, leucine, valine, and if we go on, right? In secondary structure, there is some sort of formation of the alpha helix, which is protein structure like this, or beta sheets, where there is a formation of some sort of beta sheets sort of structure. So thus the name beta sheet, or turns or loops, where it only projects some of this sort of structure, right? And then there's a quaternary structure where like a total three-dimensional structure is there, that is for tertiary, and in quaternary like multiple of these 3D structure comes together to form a one single protein, and that structure is called quaternary structure, right? So in today's video we'll be discussing about the alpha helix, beta sheets, as well as turns or loops. So the number one is alpha helix. So if you know about the DNA helix structure, it is the same sort of structure, but it is in case of the protein, right? So it is rod-like structure. Rod-like structure. So it involves only one polypeptide chain. Only one polypeptide chain. So unlike DNA, so where it can have like two or three. So in case of protein, only one sort of peptide is there, which actually form this alpha helix. So in this, what happens is, so if this is our alpha helix, right? So the R group will stick outside, right? And the CONH group, right? COONH group are going to form this bond. Okay, so it will be something like this: C double bond O and H, and this sort of structure we are going to witness. Okay, and the R group is going to stick outside. So in alpha helix, so R groups stick outside of the chain, So this help in case of attachment or interaction with other secondary structures. So what is happening is, so there is a backbone structure here, right? So where CONH sort of structure is happening here, right? So where the whole polypeptide chain is there and the R group is sticking outside. Now this R group can form a bond with another of this alpha helix sort of structure, and thus they can interact with either the beta sheets, loops, or in simple terms any sort of polypeptide. For the alpha helix, so it has a rod-like structure. So if you have seen that telephone wire, right? So old telephone wire, I'm not sure like how many of you have seen. So where it is something like this, right? So this sort of structure is basically your alpha helix, and it involved, like I said, it involved only one polypeptide chain, and the main chain, right? So the main carbon chain is on the inside, and it is stabilized by hydrogen bond. Like I said, stabilized by hydrogen bonds. So here these structure are stabilized by this hydrogen bonds, right? It has N+4 structure. So what does that mean is, so first amino acid is bonded with hydrogen bond of fifth amino acid. Okay, so for example, if this is the structure, right, and there is C O and then there's NH, right? So if these are bonded together, so this one amino acid is bonded with this fifth amino acid, and in between there are 2, 3 and 4 amino acid here. Okay, so you do not need to remember the exact structure, but you must know that there is N plus 4 structure that occurs in case of the alpha helix, right? And it is like 3.6 amino acid per ton, or around 4 amino acids. Okay, so it takes almost 4 amino acid for one turn to happen. So this turn happened because of like 4 amino acid. Okay, and the pitch of the helix is around 5.4 angstrom, right? So one turn contain 0.54 nanometer of distance. So distance from here to here is 0.54 nanometer, and the number of amino acid it takes from this to this is around 4 amino acid, and if you have to take it for like exact number, so it is around 3.66 amino acid per turn. Okay, this is important because sometime question may come. So for example, if we have 30 amino acids, how many turns would be possible for alpha helix? Or what is the total pitch of these 30 amino acids? Okay, so you need to solve this. So per 4 amino acid, the total pitch is around 0.54 nanometer. So you can easily calculate for 30 amino acid how many it will be, right? And for 4 amino acid, it is taking one turn. So for 30 amino acid, how many turn will be possible? So this calculation you have to do and you have to comment. So that I know that you guys are really studying, right? And one other thing for the alpha helix is there is overall dipole moment. Dipole moment. So it is usually from N terminus to C terminus, and the C terminus is negatively charged, and this helix can be either right-handed or left-handed. Okay, so for example, it can be like this or it can also be like this. Okay, so alpha helix can exist in both the form. Now, what are the factors that affect this alpha helix? So although all of this alpha helix is stabilized by hydrogen bonding, so disrupting hydrogen bonding can actually affect the overall alpha helix structure, right? Also, bulky amino acids are unfavorable, and it creates steric hindrance. Okay, proline is specifically unfavorable because of like bulkier group, and glycine is also unfavorable because it creates bends. So glycine is also unfavorable because it creates bends or loops in polypeptide structure, which actually disrupt this alpha helix. So the bends or loops are required in case of this, so for example, if this is a rigid structure, right, and again it is a rigid structure because it is a road like structure, right? So if we create a turn or loop like this, so there will be a hindrance, steric hindrance is going to happen with this amino acid to these amino acid. So thus glycine is also not favorable because it again bring back all of this steric hindrance towards the main alpha helix chain. Comes the part beta sheets. Like discussed, alpha helix only involve one polypeptide chain, right? But beta sheet do not follow the same structure. Okay, so it can have like two or more polypeptide chains together. So due to which what happens is, so for example, if we have a structure like CONH and then there is another CONHC. Oh wait, so there should be one C here, right? So these sort of structure can exist. So this is one polypeptide chain, this is another polypeptide chain, and this is the third polypeptide chain, right? And they can again hydrogen bond and create this sheet sort of structure, right? So this structure is called beta sheet structure. Okay, so usually there is an N terminal and C terminal to it, and in between the whole polypeptide chain occurs, right? So beta sheet consists of beta strands. So this one strand is called beta strand. Okay, so when it attaches with like multiple of these beta strands, so it forms beta sheets. Again, so there is either interchain or intrachain formation that occurs, and a beta strand is a stretch of 3 to 10 amino acid. Okay, and it is completely extended. So just suppose if you have a primary structure, right? So it is the same primary structure, an HCR group, right? And CR group here, then again COO. So this sort of structure is your beta strand. So when it attaches with another of the beta strand, they create the beta sheet, and in between there is again hydrogen bonding that is happening. Okay, so they are also stabilized by hydrogen bonds. So R groups sticks up and down like I have made here, right? So one R group is here, one R group is here, so another R group will be up and will be down. So usually it alternates, and it actually helps in better hydrogen bonding. So there are parallel beta sheets, and then there are anti-parallel beta sheets. So these are small questions that might comes during your MSQ preparation. So nobody is going to ask you exact structure of the beta sheets. But what happens is, so in parallel beta sheet, what happens is, so if the N terminal starts here, right, and this sort of structure is existing, so C terminal will end here, right? So another polypeptide chain will come, and it will have a same sort of structure. So these sort of beta sheet are called parallel, because the N terminal are parallel to each other, N and C terminal are parallel to each other. In another case, what will happen is, so there will be N structure C, right? So there is this, and then there is another group like this. So this sort of beta sheet is called anti-parallel. I hope you get it. If not, do watch the lecture again and start reading. So by reading, you are going to get most of the idea what parallel and anti-parallel actually means. And these sort of beta sheet are usually found in spider webs. Okay, spider webs. And the silk is also made of beta plated sheets. So basically, the beta sheet actually forms the silk. Now comes the loop or turns. Loops or turns. So these are small regions. So these are required at very small regions where bending is required. So what the bending does is it can create the protein part flexible, and it can help it move like in a certain direction, right? So for example, if I need some sort of bend here, right? So here the amino acid that I'm going to use is either the glycine or proline. Okay, and these sort of loops reverse the direction of the main chain. So for example, if your NC chain is going like this, so by adding like proline or glycine, they can literally flip the amino acid chain and it can create structure like this, right? So here mostly it is called loops or turns, where mostly glycine is used. So what is the advantage of this like loops or turns? It actually connects region of more regular secondary structures. So for example, we just studied about the beta sheets, right? So let's suppose like this structure is going on of the beta sheet, and now there is a turn that is occurring here, right? So again beta sheet can form, right? So N terminal is here, C terminal is here, right? So this turn is actually helping this to get back to like this direction and can form anti-parallel beta sheets. The alpha helix is usually is symbolized by this sort of structure, right? But your beta sheet is usually shown like this with certain direction. So for example, if this is the N terminal and this is the C terminal, so beta sheet will be shown by this arrow. My arrow is bit like bad. So this is again the N terminal starting here and the C terminal. So this is also the symbol for beta sheets. And for turns, so for example, if this is one alpha helix, this is another alpha helix, so turn will be shown like this U shape of structure, right? So these are called turns or loops. Turns or loops. Same case here. Okay. So another structure is called the tertiary structure, where all of like secondary structure integrate and gives you the tertiary structure, right? So here it includes alpha helices, beta sheets, turns and loops. It has amino acid side chains and even prosthetic groups, which we discuss currently. So what happens is some amino acid can interact with metals, small molecule, or any other organic or inorganic compound. Organic or inorganic compound. So those are usually called the prosthetic groups, right? And all of this creates the tertiary structure. So like we discussed, so tertiary structure, it can be the globular or it can be fibrous. And now comes the quaternary structure. Where multiple of tertiary structure come together to give us this quaternary structure, and these are usually called oligomers, right? Oligomers, or sometimes one single protein is also made up of this, like a quaternary structure. So here, like one protein like this, another protein like this, and another protein like this is creating one functional protein. So it includes all the elements from like primary, secondary, and tertiary structure to form this quaternary structure. So there is like not much of the thing that I can relate, but I can say. But the thing that you need to remember is, so these protein that are coming together either can be same or different, right? So if same sort of proteins are coming, these are called homo-oligomers, and if these are different, these are called hetero-oligomers. And interactions of this are both hydrogen as well as hydrophobic interaction, right? There is no covalent interaction that occurs here. No covalent interaction. That you need to remember. This is very important. So there occur no covalent interaction. So if that is a covalent interaction, then that protein is not the quaternary protein. With this, we are going to end this lecture here. Thank you for your kind attention.